Enzymes. What are enzymes? Enzymes are biological catalysts produced by living cells which catalyze the biochemical reactions in living organisms. Okay, so they are biological catalysts produced by living cells. Okay, which catalyze the biochemical reactions in living organisms. Okay. Example, in the case of digestion of food, enzyme is used. Okay. There will be no life without enzyme. Okay. And the prosthetic group necessary. Prosthetic group means prosthetic group. Prosthetic group means they are specific non-polypeptide unit required for the biological functions. Okay. So the prosthetic group necessary for enzymatic activity is known as coenzyme or cofactor. Okay. If the cofactor is an organic molecule, they are called as coenzyme. And if the cofactor is metals like Na plus, K plus, potassium, sodium, etc., they are called as metallo enzymes. Okay. And enzymes are globular proteins. Okay. Next, there are about 3000 enzymes are identified. Okay. And enzymes are having, for a particular reaction or a particular substrate, there must be a specific enzyme. Okay. And also they got the name from the class of the compound from which they are produced. Let us take one example. Example, hydrolysis of maltose. Here the enzyme used is maltase. See the reaction C12H22O11 on hydrolysis. Enzyme used is maltase. Okay. Enzyme is maltase. So here the enzyme is maltase, and maltase, the name of the compound ends with ASC, of the enzyme ends with an ASC. Another enzyme is oxidoreductase enzyme. Oxidoreductase enzyme. In this case, the enzyme catalyzes both oxidation and reduction reactions. So they got its name. Oxidoreductase enzyme. That is oxidation. They catalyzes oxidation and reduction simultaneously. Okay. Okay. Now, only a very small amount of enzyme is needed for the biochemical reactions. And the enzyme, next to mechanism. This enzyme used reduces the activation energy of a particular reaction. Reduces activation energy of a particular reaction. Okay. So, in the presence of an enzyme, what will happen? The formation of the intermediate will take place at a very low activation energy, and reaction takes place at a new path. Okay. Take one example. Example, acid hydrolysis of sucrose. Here the enzyme used is sucrase. Okay. So the activation energy required is 6.22 kilojoule mole inverse in the absence of sucrase. Okay. But in the presence of sucrase, only 2.15 kilojoule mole inverse. This much energy is only needed in the presence of sucrase. Okay. This much about the enzymes.